If you're anything like us, you've watched a science fiction movie, maybe something like Star Trek or Guardians of the Galaxy, and you've seen all these incredible alien life forms.
Oh, yeah, they're blue, or they're covered in scales. Maybe they have 6 eyes.
Exactly. But for all their strange looks, they almost always rely on two things that are, well, very familiar, very earthbound.
Carbon as their backbone and water as their solvent. It's the ultimate bias, isn't it? We call it carbon and water chauvinism.
Huh. I like that.
Yeah.
We're just assuming everyone is like us.
We search for life based on what we already know. But if the universe is as vast and strange as we think it is, the chemical possibilities have to be, staggering. We have to get past this idea that life has to be a carbon copy of us.
Okay, let's unpack this. Our mission today is a deep dive into the chemistry of the truly impossible, or what we thought was impossible, life that doesn't need water or even carbon.
And we're going to be pulling from some really cutting-edge research in astrobiology and chemistry to figure out what truly alien life might look like.
And most importantly, how we would ever even spot it.
That's the key. This whole investigation informs every single mission we design. If we're only looking for Earth 2.0, we might be missing the most common form of life in the galaxy.
We could be looking right at it and not even recognize it.
Precisely. So we need to seriously consider candidates like silicon or life that could thrive in, say, liquid methane or even sulfuric acid.
Right. Before we throw out the entire rule book, let's just quickly touch on why carbon is the gold standard here on Earth. Why is it king?
Well, carbon is king for 2 huge reasons. First is its geometry. It's tetravalent.
Meaning it can form 4 bonds at the same time.
Right. It has 4 connection points, and that lets it build these incredibly complex 3-dimensional structures. We're talking about the huge polymers that make us who we are: DNA, RNA, proteins.
So complexity is number 1. What's number 2?
Stability. The bond between 2 carbon atoms, the CC bond, is really strong. It's about 356 kilojoules per mole.
Which, in plain English, means it's sturdy enough not to fall apart if you cook it or freeze it.
Exactly. But it's also flexible enough to break and reform during metabolism. And here's the real kicker you mentioned earlier: oxidation. What happens when carbon reacts with oxygen?
You get carbon dioxide.
COCO.
A gas.
A gas.
And that's a massive metabolic advantage. Any biochemistry has to deal with its waste products, and being able to just exhale your waste as a gas is, well, it's ideal.
Okay, so if we look at the periodic table for the next best thing, everyone's eyes jump right below carbon to silicon.
The closest contender, yeah. It's also tetravalent, so in theory, it could form those complex chains.
But you've pointed out that in an environment like Earth, silicon just fails spectacularly. What are its fatal flaws?
It runs into 2 massive roadblocks. First, that stability issue. The silicon bond is a lot weaker, only 226 kilojoules per mole.
Fragile.
Much more fragile. And that weakness means that long silicon chains are really prone to breaking apart in water. So the process is called hydrolysis. Water just rips the chains to pieces. So, rule 1 for silicon life: avoid water. But that still leaves the other problem, right? The oxidation problem.
And that's the big one. The real metabolic deal breaker. If a silicon-based organism breathes oxygen, it produces silicon dioxide,
SiO2.
which is sand, quartz.
Exactly. Think glass. So if this thing were to exhale, its lungs would instantly fill up with basically tiny bits of glass.
That would shut down metabolism pretty quick.
Immediately. That's the silica problem. So for silicon life to even be a possibility, it has to exist somewhere completely different. Somewhere without water, and somewhere that can solve that silica problem.
Sounds like you need a whole new kind of world.
And that is exactly where the cutting-edge research is going. A huge 2020 review by Petkowski, Baines, and Seeger really shifted the focus. They started looking at non-aqueous solvents.
So not water, something else.
Something really else. They showed that silicon has a really rich and stable chemistry in concentrated sulfuric acid.
Sulfuric acid. That sounds like something you'd use to dissolve life, not create it. I know, it sounds crazy, but it's the key. In that solvent, silicon can do something called forming a hexacoordinate structure.
Okay, that's a mouthful. What does that mean?
It's vital. Carbon usually bonds to 4 things, right?
Yeah.
In sulfuric acid, silicon can actually bond to 6 surrounding atoms. This completely changes its geometry.
It makes it more complex, more stable.
It gives silicon the flexibility and structural richness that it normally lacks. It's a total game changer, and it's why people are suddenly looking at the clouds of Venus again.
Which are full of sulfuric acid droplets.
Wow.
And here's a fact that just blows my mind. This isn't all just theory. We've already done experiments in labs right here on Earth using directed evolution to force our own carbon-based enzymes to build carbon-silicon bonds.
That's right. Cytochrome C.
Our own biological machinery, when you push it, can already manipulate silicon, the element we thought was impossible for life.
It's incredible.
It really is. Okay, so from the scorching acid clouds of Venus, let's go to the complete opposite extreme, the ultimate deep freeze. Saturn's moon, Titan.
A natural laboratory for non-aqueous life. We're talking liquid methane and ethane lakes at, what, minus 179 degrees Celsius?
So the chemistry there has to play by totally different rules.
Fundamentally different. Just think about a cell membrane. On Earth, we use lipid bilayers, fats, basically. They work perfectly in water.
But on Titan.
They'd either freeze solid into a brick or just dissolve into the methane lake. Useless.
So life on Titan needs a completely different kind of container. What's the solution?
The solution was modeled by Stevenson and his colleagues back in 2015. They came up with azotosomes.
Azotosomes.
They're hypothetical cell membranes made of small organic nitrogen compounds, specifically a molecule called acrylonitrile, which is actually really abundant in Titan's atmosphere.
So they're building their walls out of something that's just floating around in the air.
Exactly. The nitrogen atoms give it just enough polarity to hold together, but the whole thing stays flexible and stable in that liquid methane. It's an elegant solution.
Okay, so we have a cell, but it needs power. What kind of fuel runs this deep frozen biochemistry? What do they eat?
The leading model is the hydrogenation of acetylene.
So they're eating hydrogen.
They are, quite literally, hydrogen eaters. They take in acetylene and hydrogen gas from the atmosphere, process it, and convert it into methane. And that reaction releases just enough energy to support life.
And if that's happening, we should be able to see the evidence, right? We should see the ingredients disappearing.
And that's what makes this so captivating. Studies by McKay and Strobel noted these strange depletions of both hydrogen and acetylene right at Titan's surface.
Chemical signature is there.
It's consistent with life sucking the hydrogen out of the atmosphere. Now, it's not proof, but it's a huge, tantalizing clue.
And that's exactly what the Dragonfly mission is going to check out in the mid-2030s, right? To see if something is actually eating that hydrogen.
That's the plan. To land right there and test for these signatures directly. The key will be to find patterns that geology just can't explain. Steep gradients, really rapid consumption in specific places.
Signs of something active, something alive.
Precisely.
Okay, so we've looked at some pretty viable, if strange, alternatives. But not every exotic life theory has held up over time. The literature has pretty much dismissed a couple of elements, right? Boron and arsenic.
That's right. Let's start with boron. It sits right next to carbon on the periodic table, so you'd think it would be a good candidate.
It has an interesting chemistry, I've heard.
It does. It can form a compound called borazine, which people sometimes call inorganic benzene, because its structure is so similar to the carbon ring. So it could theoretically mimic an organic backbone.
So why the dismissal? What's the problem?
Two really big ones. First, its preferred solvent is liquid ammonia, which is just not as common as water or methane.
Yeah.
But the real nail in the coffin is its rarity.
There just isn't much of it out there.
Exactly. Boron is way, way less abundant than carbon or silicon in the universe. So for life to spontaneously emerge from it is just statistically, well, extremely unlikely. Not enough raw material.
Okay, so boron is out. Let's talk about the really famous one, the arsenic life story.
Yeah.
I remember the headlines back in 2010.
Oh, everyone does. It was a massive story. Researchers claimed they'd found a bacterium, GFAJ1, that could swap out phosphorus in its DNA backbone for arsenic.
Which would have been proof of a completely different biochemistry right here on Earth.
It was a thrilling claim, and it immediately brought intense scrutiny, which is exactly how science is supposed to work.
So what happened? How did it get refuted?
It came down to independent replication. Other labs tried to repeat the experiment starting around 2012, and they just couldn't. What they found was that the bacteria weren't using arsenic at all.
So what were they doing?
They were just incredibly good at scavenging phosphorus. They were amazing survivors, able to find and use the tiniest trace amounts of phosphorus while ignoring all the arsenic around them.
And the original study was just flawed.
Yes. The follow-up study showed there were issues with contamination and washing protocols in the original experiment. And it took a while, but the final word just came down. In July of 2025, the journal Science officially retracted the original paper.
Wow.
Yeah. The arsenic life case is now a landmark example of scientific self-correction. It's a decisive refutation of arsenic-based DNA.
Okay, so we've established that truly alien life could be weirder than we can imagine, based in sulfuric acid or liquid methane. So we can't just look for Earth-like signs. How do we look for any life?
We have to rely on biosignatures. A biosignature is any pattern that is best explained by life, no matter its chemistry. NASA even has a formal system for this, the ladder of life detection.
Which basically says you have to rule out every non-biological cause first.
Every single one. And for exoplanets, where we can only look at their atmospheres from light years away, the strongest biosignature we hunt for is something called redox disequilibrium.
That sounds complicated.
It's actually a simple idea. It's finding two gases in the same atmosphere that should have destroyed each other. They're chemical enemies.
Like finding lots of oxygen and lots of methane at the same time.
Perfect example. On a dead world, they'd quickly react and neutralize each other. But life acts like a planetary battery, constantly pumping both out, maintaining that imbalance on a scale that geology or sunlight just can't keep up with.
It's a sign of a system that's being actively driven by something.
Exactly. We also look at surfaces. On Earth, we have the vegetation red edge.
Where plants reflect a specific kind of infrared light to avoid overheating.
Right, so we look for similar, sharp, strange spectral features on exoplanets. Maybe their life is purple or black, but it would likely still evolve some kind of pigment to manage energy, creating what we call a biofabric on the surface.
So for planets far away, we're looking for weird atmospheric cocktails and strange colors. But for places we can actually visit, like Mars or the icy moons, we can send rovers.
And rovers can dig. They can look for in situ signals, things protected from radiation. A key one is looking for an enantiomeric excess.
Okay, another big term. Break that down for us.
It's about the handedness of molecules. Think of your hands. They're mirror images, but they're not identical. Many organic molecules are the same. They come in a left-handed and a right-handed version.
And non-biological chemistry makes both.
Always. A 50-50 mix. But life on Earth is incredibly picky. It almost exclusively uses left-handed amino acids. So if we go to Mars and find a big pile of organic molecules that are all, say, right-handed...
That's not random. That's a sign of selection, a sign of biology.
It's one of the strongest possible signatures you could find.
And finally, we get to the ultimate sign of organization, technology. We're not just looking for microbes, we're looking for technosignatures.
Right. We're looking for signals that simply can't be natural. This falls into two big categories. First, direct communication.
Radio signals or lasers.
Yeah.
The classic SETI search.
Yep. Looking for narrowband radio signals or continuous lasers that stand out from all the cosmic noise. And second, we look for planetary scale engineering.
You mean alien artifacts?
In a way. Things like weird industrial pollutants in an atmosphere, artificial lights on a planet's night side, or, the Holy Grail, the transit signature of some kind of megastructure, something huge and artificial blocking a star's light.
But in the end, no single one of these is a smoking gun.
Absolutely not. NASA's Confidence of Life Detection Scale, or COLD scale, requires multiple independent lines of evidence. You have to find the weird chemistry, the molecular patterns, the atmospheric imbalance, and rule out every other possible cause until life is the only explanation left.
So after this incredible deep dive into alien chemistry, what are the key takeaways for you?
I'd say there are three. First, carbon is king for a reason, but silicon is viable. It's a real possibility for life, but only if it finds a very special home, like concentrated sulfuric acid, which lets it bypass that whole exhaling glass problem.
Second, that life on deep freeze worlds like Titan is theoretically sound. It just needs exotic adaptations, like using methane as a solvent and building its cell walls from nitrogen compounds, those azotosomes.
And finally, that detecting any life means we have to think beyond ourselves. We have to hunt for universal biosignatures, things like that chemical imbalance in an atmosphere or that telltale handedness in molecules.
It's so easy to just picture aliens that look like us, but the universe is almost certainly far stranger. And our missions, like the one heading to Titan, are finally being designed to look for those hydrogen-eating pathways.
It makes you wonder. We know we can force life in a lab to manipulate silicon. So given how vast the universe is, what about an element that's rare here, like boron? Where might it find that perfect, cold, ammonia-rich world it needs to thrive? The chemistry is possible. The universe just has to be big enough. That's something for you to explore.